Hey everyone! Speaking with us today is Dr. Adam Arkin, Professor of Bioengineering at UC Berkeley and Senior Faculty Scientist at the Lawrence Berkeley National Laboratory. Dr. Arkin, would you like to tell us a bit more about your background and research?
Sure, Lydia, thanks. So, this is Fast and Furious. It's meant to give you a sort of an overall sense of what we do. I'm sure a lot of this will be too complicated, but I hope you'll get a basic idea of what we're trying to accomplish here. So, my field is something called systems and synthetic biology, which means I try to understand how organisms work as integrated systems of molecules and cells and organs and in contact with their environment. And I try to do that using detailed measurements and modeling. What synthetic biology means is that once I understand that, I try to then create new function in cells to solve problems that we think are a good idea to solve. My particular bent is trying to understand how to engineer organisms for extreme and new environments and environments that are generally outside bioreactors unless they happen to be on Mars. And so I want to tell you a little bit about how we think about this. One second. Okay. So my laboratory is a relatively large laboratory as academics, though. We have around 50 people or so, but I work with hundreds of people because I tend to work in very large programs that I design. There's 4 programs that I basically run. One is called Enigma, it's this one over here, the symbol over here, where we try to go out into the field and understand how things work in the external world, in particular in a field that I'll describe a little bit later, but it's outside, below ground, in the earth. I have another program which is about how to take the organisms that we find and engineer them to do new things. I have another program that takes all the data we generate out here to work with myself, my lab, other scientists in the world to figure out how biomolecules function, how they work together, and then make models of how they can operate in the environment and how we might engineer them. And then this program is Cubes, which is how to build a manufacturing plant to support a family of astronauts living for a long time on Mars, with the idea that this manufacturing plant has to be completely sustainable. It has to be completely closed loop. It can't make any waste. It has to use only what's available on Mars. We can't afford to bring stuff with us. So whatever we develop for Mars should have applications here on Earth for sustainable biomanufacture of food, pharmaceuticals, and building materials. Okay, so let's talk about what this is about. Really, this is about how to figure out how to engineer cells for living in new environments. Now, the cells I tend to engineer are bacteria or fungi, some plants from time to time, but mostly bacteria and fungi. And bacteria and fungi are everywhere. To give you some sense of things, and viruses, I do viruses as well. So there are about 10 to the 30th bacteria on Earth. That's more than the stars in the sky. It's more than you can possibly imagine. There's about 10 to the 6th, a million of them in a gram of soil, and 10 to the 5th different species of bacteria in that same gram of soil. There's around 1 billion or 10 billion phage, these little viral things that can attack these bacteria in there. So they're living in this very complex, environment. In your gut, it's even denser than that. And then you have different communities on your skin, and your armpits, and your nose, and your mouth, and so on. And so we're interested in how these bacteria assemble in these different locations, how they process those environments, how we can get a handle of them to do the job. Now, nearly every electron that is hit by light from the sun passes through microbes before it gets to us. They are the main way that we process energy in the universe. And so again, they're the major handle by which we can change the flow of carbon, nitrogen, phosphorus, and sulfur flow from the world. So we'll talk a little bit about our work in contaminated watersheds, contaminated humans, and in building manufacturing plants. We'll start with Enigma. So Enigma is trying to solve a particular problem. This particular problem has to do with the contamination of the Earth by nitrate and uranium. That might seem pretty weird, but we use nitrate to fertilize our crops. In fact, the process in which we create that fertilizer, that nitrogen-based fertilizer, is called the Haber-Bosch process. It uses a significant fraction of the energy on Earth to make this happen, to support our populations. Because we use so much of it, we've contaminated the subsurface of the world. In the United States, this is where we can see the highest contamination here. And what you're seeing here, of course, is the California agricultural belt, where we do all our agriculture. And this happens to be the aquifer that feeds. The aquifer is a catchment of water that feeds all our crops in our breadbasket. And we have contaminated it with very large amounts of nitrate that are poisonous to us. Now, when nitrate is there, it is acidic and it leaches minerals out of the soil, and it creates all this uranium, because we're a uranium-rich country. It also is there for the first time Kansas farmers are pulling up uranium in their crops. So we would like the microbes to recover that nitrate, to detoxify it, and move it to the plant so the plants can use it more effectively. Now, we actually don't work in the agricultural world, though. We work in a place where we were pretty bad. We were making nuclear weapons and doing nuclear processing. This is a classified site called the Y12 Site. It's out in Oak Ridge, Tennessee. And a lot of people still work there. And we have contaminated this watershed with a bunch of uranium. And in fact, we work right next to this parking lot. Now, this parking lot is sitting right on top of a large amount of waste uranium. And when the cars run onto this parking lot, they pump the uranium and out it comes, and it contaminates the watershed. And we've been mapping this watershed with extreme precision. So we can, for example, tell you, they put these monitoring locations all along the edge of this pond where the stuff is leaking out, for example. And we can measure, for example, all the geochemistry, all the soil sediment geology, and all the hydrology at that site, so how the water is moving and in three dimensions, and what that soil and sediment looks like. We can then drop these wells down and measure in real time things like the pH, 53 different metal concentrations, carbon and nitrogen, things like this. And we can measure who is there. This is actually just a plot which shows where every line is a different depth, and every column is a different species. We can ask, where is that species in depth? So we get these communities that form at different depths. And then we can ask what they're doing. So we can figure out what genes they have, what things they can do inside themselves, and therefore figure out what each community is contributing to the change in the mineralogy of all this site. So now that we have that, we can bring them into labs. We can map. This is what's called a phylogenetic tree. And what we're seeing here is this is a calculation of the evolutionary relationship of all the organisms, all those species that we're observing there. We can see they're an extremely diverse group. So humans would be a single point all together on this graph. This is representative of 4.5 billion years of evolution, where we have all these organisms that we observe at this location. And then these are the ones, these black ones are the ones we're able to get into the laboratory to begin to study them. When we have them in a lab, we can measure things about them. So this is, for example, all these different organisms on this axis, and then all the different metabolites that they create or they eat when they're dropped in a given condition. So we can measure how they're transforming the environment by what they eat and what they secrete. And we can ask how well they grow in those same environments. So how well are they using those nutrients? So we get measures of efficiency and transformation. And then we can ask how every single gene plays a role, because we've built these genetic tools that allow us to turn on and off every gene in that organism. And then we can measure how these properties change as we turn it on and off every gene. And we've done this for now tens to 100 organisms that we've come from this site. So now we have a gene to organism to its ability to operate, and we'll be able to map that organism out into the world. And we can begin to ask how they operate as communities to do their job, molecule per molecule, gene per gene. Then we can put them together into communities in the laboratory that simulate what's going on in the world, and we can measure how they actually operate in the lab as communities. In this particular case, we can map exactly how this pair of microbes reduces nitrate. However, just because we can measure all this doesn't mean we can understand it. So we have to put all this information, all this data that we have, gene per gene, molecule per molecule, everything, into complicated models. Now, one of the things that we've done, though, is that while we've collected these organisms, we've discovered interesting things. This, for example, we discovered in this particular environment an organism. Let's see if this will work. This is a little device we have that allows us to measure organisms with extreme precision. Each one of these little rods here is a microbe, and what you're seeing them is them dividing and being pushed out of these tubes. And we have this artificial intelligence that can tell us where every cell is. It can quantify every cell's behavior. how it grows, and what it's making. Because this little white, these are the same image, this one here and here. This one in light, this one as it emits light. And this light emission is where it's making plastic, it turns out. So these guys we found in the subsurface make a natural sort of plastic. And we're asking how they do that here. We've engineered them. We figured out these are organisms we should engineer to make new plastics that we'll use in our space program later. But we have this extreme precision of measurement of these guys, and the extreme ability to engineer them to make these sort of bioplastics. I won't discuss this today. Too long. We have other engineering we do. So we put all this data, all these measurements into this thing that I built called Kbase. So Kbase you can use Kbase. It's an open source, open access system that gives you complete access to DOE supercomputing and to all this machinery for data analysis and modeling that we put into it. And it allows you then you can integrate your own tools, and it will reason on your data to do new things. So we have around 500 different things you can do to data, and people who use this follow like thousands of different paths to how to analyze this data. This is how our users use the system. And they speed a lot. They cover lots of things. They kind of take all this sequence data. We have to use genomic sequencing to figure out what's out in the field.
Here's some information.
It can allow you to assemble those sequences into genomes. It'll annotate those genomes. It'll figure out how the genomes relate one to the other. It'll then make models of each genome, and then it will make models of the groups of genomes together. So what's really neat about this from the point of view of how we think about this socially is that the way the system works is when you do your analysis, it writes a paper for you as you do it. You can actually publish this paper directly from the system. This is how an analysis from our system looks when published out with a single button click. Now, you guys can actually learn how to use the system. We have an educator program that has 144 member educators from across the world. They will go from high school all the way through postgraduate work. You can access these, many different ways of doing all of this stuff, and the data, and you can access other people's data to use it. And you can learn about these more here at these links. We also have lots of video tutorials if you want. You can go to YouTube and look them up on how to use them. But this is completely open access to you. You can sign up and use it immediately. Now, we've used it in this particular case for the data I showed you above to begin to build models of our system. So what we're showing here is that our system is able to look at, infer all the networks of interaction. So each one of these dots is a metabolite. Every little arrow is a reaction that can occur between the metabolites. And our system has inferred how these organisms are transforming the world through all these chemical reactions. We then are able to fit this model of the world to all that data I showed you before, meaning to make sure that they're consistent. This just shows you how we do it. I'm not going to belabor it. It's all these different functions that we use to make this happen. Then we can predict things like, as the environmental nitrate increases, which organism is doing which job. We see that together, as the nitrate increases, this community increases its growth, but that in fact initially is Rhodonobacter, which is this particular microbe, which is doing all the nitrate reduction, but at a critical location it ends up being a Pseudovorax, another microbe which does it. So we're able to predict exactly who's doing what when under what conditions. So now that we have this sort of control and prediction, we can even use this in design mode. And so we have this mission we do with NASA trying to find integrated biomanufacturing for Mars. So the idea here is really engineering for the next planet. We're thinking about how to make these very contained environments where you can live with no impact on the world around you, but be fed, get your medicine, and make more building materials on demand so you can build out your environment as time goes on. The way that we do it is we've made an inventory of everything that exists on Mars that we can use and regenerate. And that includes carbon dioxide, nitrogen and oxygen, minerals from the sediment and the like. And we figured out how we would harvest that using various simple machines and then push them into these reactors. Now, these reactors are all things that are driven by either plants or microbes that can take those materials and turn them into what we want, either plastics, like I showed you, medicines, or food. This requires us to think through all these different engineering disciplines. And our teams are very integrated with people who build machines, who engineer plants, who are nutritionists, who are material scientists, who are physicians, and so on, trying to figure out how to put all this stuff together. I'm just going to say that it's very, very integrated. We've defined an entirely new field in which we were launching a program where you can get trained in this and how to actually engineer for this sort of environment. Now, I'll just give you an example. This is an organism called that we're engineering for this purpose. This is called Arthrospira platensis. It's also called spirulina. You can buy it at a health food store. This is a bacterium you can just eat. It has a very, very high protein content. It looks kind of ugly. It forms these, on the plate. Oh, beautiful. I'll give it a like it. It forms these filaments that are motile and will move around. And we've been able to figure out how to engineer these guys. They're very hard to engineer. And we can engineer them to glow, for example. This is them glowing on a plate now. You can see them very, very up close. These are guys we've engineered to glow. But what we've recently done is engineered them to make Tylenol. So now you can have this organism which you can grow in your jar, and without anything other than light and water and nitrogen from the atmosphere, it will create Tylenol for you that you can just eat. You take it right out of the jar and eat it, and it will give you a dose of an analgesic, like Tylenol. So we've been able to put this all together, and we have systems engineers, things that harvest carbon and nitrogen from the atmosphere and turn into sugars for the other organisms. We have plastic making systems. We have people who are making engineered lettuce that make human biologicals and are engineered for high nutrition. We have the spirulina, for example. We put it all together into a series of bioreactors that can reuse the waste that they produce. So you think of this as an integrated system for building this Martian city of the future and making these distributed manufacturing plants that you could have in your backyard to feed your family for a year, for indefinitely, and give them medicine and building materials with just living in the world and using your waste. So I'll just stop there. I'll say that's what we kind of do. It's built with large teams of people. One of my privileges is to belong to the National Laboratory, where team science is very celebrated, and we do a lot of work there. And this gives you some sense of the size of the teams that we're working with to get this done. My own team is much smaller, but there they are. There's an old picture of them. And with that, I'll stop and I'll take questions. Thanks.
Hello, Professor Arkin. Once again, thanks so much for coming here and presenting here for us. So we'll move on to the questions now. So first question we have is, what motivated you to educate aspiring students and become a professor?
Well, it was kind of interesting. Honestly, the reason to become a professor, if you're not, even if you're not thinking about educating first, is that it's one of the academy, the university, is one of the few places that you and people that you work with can together think about new clever ideas and develop them like an artist. We're not driven by a bottom line, we're not driven by profit or economics. We're driven by what interests us. And so the university is a place that you can follow your interests. You want to educate because young people are full of new ideas and energy. And they come and they're excited, and they can come and work with me, and they can invent new things. And I can't do that all myself. There's too many moving parts here. And the only way you can innovate is by having young people do it, but they don't know how to do that to begin with. So you have to teach them how to think this way and how to use the different tools. And so it becomes part of your mission as someone who wants to build new ideas, to make sure that you have a workforce who can join you. And so that's what drove me to become a professor, because I could be at the National Lab and just do research, but I have to teach people to think the way I want them to. And so the university lets me do that. And then this sounds like it sounds sort of silly, but you learn a lot by teaching people, both because you have to organize your thoughts and because they have new ideas and new questions you haven't even thought of. And so they teach you too. So it's a perfect situation.
It's a very interesting outlook on teaching and becoming a professor. Our second question is on bioplastics.
Mm-hmm.
How do the uses of bioplastics vary from regular plastics?
Yeah, so it's a really good question. So the reason why we like bioplastics is for two reasons. The first reason is that instead of having to use oil, which is the major way that we make plastic now, we can make that out of air. out of CO2 and nitrogen, which is a much more regenerable resource. So we can actually sequester carbon from the atmosphere to do this, thereby, impact. But the best thing about it is that the plastics that we make with bugs can be eaten by bugs. So it means it can be recycled directly by the organisms that make it. And so now we have a closed loop design. We're not using a non-renewable resource to make the plastic, and we're able to reuse the plastic. It completely breaks it down and can be made into a whole new plastic, a whole different type of plastic. So it's really, really powerful to be able to do this. Now, honestly, we are not at the point where this is economically viable for the scale that we want. But that's what we're moving towards now, and I think it's a huge open field. Very exciting.
How do you start that loop though? Like, what do you feed the bacteria at first so that you create the first piece of plastic and then it can be recycled?
Yeah, really good. Another really good question. So first things first, so nearly everything has to eat a few fundamental atoms to survive, right? It needs carbon, nitrogen, oxygen, phosphorus, and sulfur, and a bunch of little other nutrients. Now, the major way that we feed organisms like bacteria or yeast industrially tends to be refined sugar. That's expensive. So like most of Brazil is made of sugar cane, and you harvest it and you make that sugar so you can have your Twinkies and they can have their sugar, right? But that's very expensive and it's actually not very sustainable. So the organisms that we're trying to use try to use other sorts of what we call feedstocks. And so those would be things like carbon dioxide, which is in the air, you breathe it out, right? And carbon dioxide is a major greenhouse gas. Or methane, for example, which is what sheep and cows burp out and cause greenhouse gas problems. Or we deconstruct waste. So things like paper, we just deconstruct it. We have it eat raw paper so that we eat it. So these bugs are engineered to eat these different types of inputs, right? And when they eat those inputs, they can then create the plastic internally by using their own metabolism. Now, there's other things you have to feed them, like for example, like we told you that we don't want to feed them nitrogen sources that come from Haber-Bosch process. That would be bad. So we want to feed them things like urine, the high nitrogen. We want to feed them things like waste products that have nitrogen in them. So that would be other sorts of solid waste or nitrogen from the atmosphere, N2. So they have to fix nitrogen directly.
So the final question is, how can high school students get involved with trailblazing research on bioengineering?
Yeah. So the first thing, first of all, there are internships out there for you to do that are formal that you can apply for. Almost, one thing I'll say is, and we've had a lot of high school students in my laboratory, and they've just written me, and it just happens to be at that moment I have someone who wants to mentor students. You have to be persistent. You got to ask like 20 times to the one person and then 100 times to all the different people before you get a hit, right? But email is cheap, so do some research, figure out who you think is interesting, write them an email 2 or 3 times if they don't answer you. And they will, and sooner or later you'll find someone who wants to take you in. The other thing I'll say is that there are summer programs specifically in biotechnology that you can apply to. They do tend to have some cost. And the final thing I'll point out, and you guys might not be aware of this, but there are open biohacker labs, maker labs in the Bay Area where you can sign up for a very nominal fee, and they will have internal classes and they'll teach you how to use their equipment, and you can go and just do biology there. It's very, very cool. And so I would suggest you look those up.
All right. Thank you so much, Mr. Arkin. Well, everyone, thank you so much for watching our interview with Professor Arkin. Take care, and we'll see you all next time. Bye.
Thank y'all.